Okay, what we're going to look at in this little video is oxygen dissociation curves. Now, before we carry on and start looking at that, obviously oxygen dissociation curves, it's all about hemoglobin and the loading and unloading of oxygen and being transported around the body. Just thought it would be worth just making a couple of points about red blood cells. After all, it's in these structures here that the hemoglobin is found. As it says there's around about 20 to 30 trillion red blood cells in the human body, and each red blood cell contains around about 280 million hemoglobin molecules, and each hemoglobin can carry up to 4 oxygen molecules. So you can see the ability to carry oxygen around the body is immense. It's always worth just brushing upon and thinking about the adaptations of a red blood cell to enable it to carry out its job of carrying oxygen around the body. It's an incredibly small size, only about 7 micrometers. Remember, 1,000 micrometers in a millimeter. So that means there's a very short diffusion pathway for any oxygen to get in. It's biconcave, it's got that biconcave shape. If we just look at it sideways, look at it like this. That provides a large surface area and a short diffusion pathway. If it was spherical like this, any hemoglobin in the centre would have this much larger distance for the oxygen to diffuse into it. Being biconcave means any hemoglobin is never far away from the membrane of the red blood cell. And finally, no nucleus, therefore there's more space for hemoglobin. Okay, so here's a hemoglobin molecule, and this is what it looks like in a bit more detail. First of all, hemoglobin is a protein. The primary structure is it's made up of 4 polypeptide chains, and here you can see them. Here and here, the 2 lighter coloured green colours, and here, the 2 darker colour chains. Each one of those is then coiled into a helix, which is its secondary structure. The tertiary structure is the fact that each of those coiled chains is then folded into a very precise shape, and you can see that there. And then the quaternary structure is that the 4 polypeptides are linked, and each one has got a heme group associated with it. Now the heme group is the bit that actually carries the oxygen. That's ferrous group, it's got iron in it, and each one of these, there's one, there's another, there's another, and there's another, can carry 1 oxygen. So that's why each hemoglobin molecule, of which here is 1, each one can carry 4 oxygen. Now hemoglobin's got a pretty tricky role in that it's got to be able to pick oxygen up in areas where there's lots of oxygen, i.e. the lungs in humans or gills in fish, and then take it to and deposit and release the oxygen in areas where there's not much oxygen, so respiring muscles and respiring tissues. Now different species have got different types of hemoglobin. Now what makes that difference is just small changes in the amino acid sequence. What that does is it gives the hemoglobin different affinities to oxygen. Now hemoglobin that's got a high affinity means that it's going to hold on to and bind to oxygen very easily, but the trade-off is it doesn't release it as easily. Whereas hemoglobin with a low affinity, it's going to be harder for the hemoglobin to pick the oxygen up, but it's going to release it much more readily. Now the best way to describe it is by 2 examples. So the mackerel, very energetic fish, very fast moving, a lot of muscle tissue, a lot of mitochondria respiring, high demand for oxygen. Therefore, its hemoglobin has got a very low affinity to oxygen. And that sounds a bit strange in that it needs lots of oxygen in it, but its hemoglobin doesn't carry or doesn't hold on to it very easily. But that's a good thing really, because that means the hemoglobin lets go of it really, really quickly. However, the trade-off is they can only live in a high oxygen area. So they can only live in well oxygenated conditions because they need that high level of oxygen to enable the hemoglobin to bind with the oxygen, because it's got a very low affinity with it. Compare that to the lugworm. Now the lugworms, there's one there, you've probably seen them as their casts that they leave on sandy beaches when the tide's out. They spend a period of time, if just under here, there'd be a burrow just under the sand there, where one of those lugworms lives. When the tide's gone out, up here, the bit of water that it's got there is what it's got to live in until the tide comes back up. Now these things along here, they're its gills, so that's how it gets its oxygen as opposed to lungs in humans. So that little bit of water that it's in its burrow, it's going to start using that oxygen up. So what it needs to be able to do is get every last little bit of oxygen out of that water. So what that means is its hemoglobin is going to have a very high affinity to oxygen. That means it's going to grab hold of it, bind with it, join with it much more readily. But on the flip side, it doesn't let go of it as readily. So that the trade-off is it's going to have a low metabolic rate. Yes, it can live in low oxygen environments, but the downside is it can't have a high metabolic demand. It can't be busy, moving, keeping warm, etc. Demands on metabolism. It needs to keep that down to a minimum because its hemoglobin does not let go of its oxygen very readily due to the fact that it's got a high affinity. So, how does this loading go about? Well, let's just imagine here we've got a hemoglobin molecule, and here's an oxygen molecule. Now, to begin with, it's very hard for the oxygen to come in because it's got to bind with one of these heme groups. It finds it hard to get in. But eventually, and remember this is just a chance encounter with a heme group, it will get in. And it binds to one of the heme groups. Now what that does is it causes the tertiary structure of this hemoglobin molecule to change slightly, and it becomes less compact. What that does, it means it's easier for subsequent oxygen molecules to join, here and here. So we've now got a situation where in this hemoglobin molecule, 3 of the heme groups have got an oxygen attached to them. The problem comes though with the 4th oxygen molecule. The 4th oxygen molecule, yes, it can come in, but it's going to find most of the heme groups having a oxygen already bound to them. So that means for the 4th oxygen molecule to join to the hemoglobin molecule, it takes a little bit longer. It's a bit more, you need high oxygen levels for that to occur. Now, I'm going to show you this on a graph. So this is the sort of curve that you need to be able to interpret, and it's an oxygen dissociation curve. On the x-axis here, we've got the oxygen partial pressure. Now the partial pressure is used instead of concentration or percentage. Because if we were to have percentage of oxygen along here, which you can imagine in some environments is a high percentage of oxygen, which is fine, but it doesn't take into account pressure. And when you're talking about gases, pressure plays a really important role as well. So 21% oxygen at sea level, relatively high pressure, is going to cause those air molecules to be slightly closer together, meaning they're going to bind to the hemoglobin more readily. Whereas 21% oxygen at top of Mount Everest, much lower pressure, means that the oxygen molecules are going to be more spaced apart, meaning they're not going to bind with the hemoglobin as readily. So we use partial pressure in kilopascals, the unit of pressure measurement, as an indication of how much oxygen there is available. So just think of it in terms of the amount of oxygen that's available. Percentage saturation of hemoglobin. Here we've got 100%, 50%, 0%. 100% saturation means every heme group and every hemoglobin molecule in every red blood cell in a particular region of the body has got an oxygen attached to it. Here's 50% and here's 0. At 0%, not one of the heme groups in any of the hemoglobin molecules in any of the red blood cells has got any oxygen attached to it. So the curve follows this very typical sigmoid shape. The reason we have this slow start at the beginning is because, that's like I said, the heme groups, the tertiary structure is very tightly packed, which means it's very hard with a chance encounter for an oxygen to bind with a heme group. So as you increase the amount of oxygen, there's only a very small increase in the amount that's saturated. Reason being, the tertiary structure of the heme group, the hemoglobin molecule, is tightly packed, meaning it's hard for the oxygen to bind with the heme group. However, once one heme group's got an oxygen attached to it, the tertiary structure changes slightly, making it easier for that second and third heme group to have an oxygen molecule joined to it. So that's why there's a steep rise here. Just a small increase in oxygen here leads to a huge increase in the amount of oxygen that's saturated the hemoglobin molecules, because the second and third molecules are much easier to join to. We then have this plateau here, and hopefully you can remember the reason that is because as the first, second and third heme groups get joined to an oxygen, it just means there's less likelihood of the final heme group getting an oxygen attached to it, because the other three available slots, if you like, have been taken. So there's slow rise, steep increase, and then this plateau. There you can see this region here, difficult for oxygen to become loaded due to the polypeptides being close together. Small increase in oxygen partial pressure causes large oxygen loading and unloading. The reason I'm saying that is if you increase the oxygen, there's a huge increase in the amount of oxygen that's loaded. If you decrease the oxygen slightly, it's going to let go of its oxygen much more readily. And here there's a plateau in the curve due to near 100% saturation. There's very few free heme groups available, meaning there's less likely that an oxygen will bind to them. Now, different hemoglobins are going to have different affinities to oxygen, and this is just shown here. Here's, if you like, a typical hemoglobin molecule. Any curve shifting to the right means that there's a lower affinity to oxygen, which means they take up oxygen less well, but they release it much more readily. So that's the macula as the example. So curve shifted to the right, lower affinity, don't bind to the oxygen as easily. That means they take it up less well, but they're going to release it much more readily. That's good for animals or organisms that have a high metabolic demand. They need lots of oxygen to be supplied to their muscles. Downside is they have to live in high oxygen environments. A shift to the left means that the hemoglobin's got a higher affinity to oxygen. It takes up oxygen really, really well, but releases it much less readily. And that's good for animals that live in low oxygen environments. They want to bind and get hold of and associate with that oxygen much more readily. However, they don't offload it, release it as easily. So the trade-off is they have a low metabolic demand. They can't be active organisms. Benefit is they can live in low oxygen environments. So if I just try and demonstrate that for you now, let's just consider this oxygen partial pressure here. What's that going to be? That's going to be about 16, something like that, kilopascal of oxygen. The blue curve here, with a low affinity of oxygen, they're going to have 5-10% of their hemoglobin saturated with oxygen. This normal, better word, hemoglobin molecule is going to have 60% of its hemoglobin bound with oxygen, associated with oxygen. And the high affinity hemoglobin is going to have 100 should be up here somewhere, 80-90% of its hemoglobin associated bound to oxygen. So being on the red curve means you've got much more oxygen in your system, but it just doesn't let go of it as readily. The blue curve here, you've got much less oxygen in your system, but it's been let go of much more readily. Now, the final thing that we need to talk about is this thing here called the Bohr Effect. Now, just let me talk you through these points here. In rapidly respiring tissues, carbon dioxide is produced. Carbon dioxide dissolves in fluids, so in blood and the plasma, tissue fluid, to form something called carbonic acid. This carbonic acid then lowers the pH of the blood. The lowering of the pH of the blood causes the hemoglobin molecules to change shape, and the change in the shape causes the oxygen to be more readily offloaded, to be more readily released. Therefore, more oxygen is available for respiration. So that's the body's way of meeting supply and demand. As respiration increases, CO2 increases, drop in pH, hemoglobin molecule changes shape, releases the oxygen more readily. That's fantastic as you're working really hard. Humans, for example, quadriceps, as you're running, you're going to build up CO2 in that tissue due to the mitochondria respiring. That's going to lower the pH in that aeric region. As hemoglobin comes into it, it encounters this lower pH blood, and then that means it's going to change the hemoglobin molecule shape slightly, causing it to release the oxygen more readily, providing those mitochondria with the oxygen that they require. And here you can see it. It's called the Bohr Effect, and the Bohr Effect is a shift of the oxygen dissociation curve to the right, which means the lower affinity for oxygen, which means it gives it up more readily. So here you can see the red curve is at a low oxygen environment, at low carbon dioxide environment. As you start to increase the carbon dioxide concentration, the curve shifting to the right, and as you increase it further still, shifting to the right even more. And the final slide here shows you the effect of that. So at a given oxygen partial pressure here, I've chosen 8 kilopascals of oxygen or thereabouts. In the low carbon dioxide concentration, the blood is still about 80% saturated with oxygen. The blood hasn't released its oxygen as readily. At a medium carbon dioxide concentration, because this curve shifted to the right, it's now only holding on to 40% of its oxygen or thereabouts. So in other words, it's released 40%. It's gone down from 80 to 40%, which is great because there's an increase in carbon dioxide concentration here. That means those mitochondria are respiring. That means they need that extra oxygen, and that's been supplied. And if we increase the carbon dioxide concentration even further, again, shift of the curve to the right, further oxygen being released, further oxygen supply to the mitochondria. Okay, so that's the oxygen dissociation curves. It's the ability of the hemoglobin to bind with oxygen in high oxygen environments, such as the lungs or the gills in fish, and release the oxygen in low oxygen environments, so those respiring tissues. You need to be able to know how different hemoglobins can be used for different organisms, and you need to be able to deal with the Bohr effect and how carbon dioxide and why carbon dioxide affects the affinity of oxygen associating with hemoglobin.